I'll start.
Just, please
Okay.
proceed.
All right, sir. Hello, everyone. Welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's class will focus on mycology, part 1, for BSc MLT first year students. Mycology. So, mycology is the scientific study of fungi. It highlights that medical mycology is a specialized field focused on understanding fungi that cause human diseases. Fungi are diverse organisms, including yeast, single-celled, and molds, filamentous. Fungi pathogens can be divided into two main groups, that is, true pathogens and opportunistic pathogens. True pathogens which can infect healthy individuals and opportunistic pathogen which primarily cause disease in immunocompromised people or people having underlying health issues. So this introduction establishes the foundation for learning how fungi impact human health. Terminologies. Here important mycological terms are defined. Dematicious fungi contain melanin in their cell walls, giving them a brown or black pigment. Dimorphic fungi can grow as either molds or yeast depending on environmental conditions. Imperfect fungi lack a known sexual stage, while perfect fungi can reproduce sexually. There's one more, polymorphic fungi have multiple independent growth forms. It also differentiates mold, that is multicellular colonies of hyphae, and yeast, that is unicellular round to oval cells that reproduce by budding. So which reproduces by budding. Understanding these terminologies is crucial for classifying fungi in clinical settings. Molds, it is nothing but hyphal or mycelial colony of growth. Yeast is unicellular or spherical to ellipsoid in shape, that is 3 to 5 μm. Fungal cells reproduces by budding. Epidemiology. Humans are relatively resistant to fungal infections, with exceptions like dimorphic fungi that can cause serious diseases. It also outlines predisposing factors that increases susceptibility to mycosis. These include immunocompromised states, that is, for example, HIV, chemotherapy, organ transplant, serious underlying illness, immunosuppressive therapy, prolonged antibiotic use, and major surgical procedures. It emphasizes that the host immunity is a key determinant in fungal diseases. Unlike bacterial or viral infections, most fungal infections are not communicable. They are typically acquired from the environment. The main routes of acquisition are inhalation of spores, direct contact with spores, or trauma inoculation into the skin. This means fungal diseases are largely opportunistic and accidental rather than spreading person to person. General characteristics of fungi. So this slide lists the basic biological features of fungi. They are eukaryotic, thallophytes, meaning they have true nuclei but lack chlorophyll and depend on organic matter for nutrition, that is saprophytic nature. Their cell wall contains chitin and their cell membrane contains ergosterol, which is a target for antifungal drugs. Fungi are generally obligates aerobes, which requires oxygen. Though some yeast can grow anaerobically, they are also more resistant to osmotic pressure, can survive in low moisture, and utilize complex carbon sources like lignin. These adaptations make fungi environmentally hardy. So it compares fungi with bacteria. Unlike bacteria, fungi are eukaryotic, have a true nucleus, and reproduce both either sexually or asexually. So they are generally larger than bacteria, grow more slowly, and are unaffected by antibacterial drugs. Their cell wall composition and cell membrane sterol are important diagnostic and therapeutic distinctions. So obligate aerobes, almost all molds are aerobic, most yeasts are facultative anaerobes. So most are more resistant to osmotic pressure rather than bacteria, as well as it can grow on low moisture content and require less nitrogen compared to bacteria. These are often capable of metabolizing complex carbohydrates such as lignin. So overall, understanding these comparison helps in why antifungal treatment is very different from antibiotic therapy. Fungi versus bacteria. So fungi include nucleus, which is defined as nuclear membrane, and in bacteria, there's no nuclear membrane present. Mitochondria is present in fungi, whereas absent in bacteria. Cell wall, fungi contain glucans, mannans, chitins, whereas bacteria contain peptidoglycan layer of cell wall. Cell membrane of fungi contains sterols. No respiratory enzymes is found in fungi, whereas in bacteria, sterols is absent with respiratory enzymes. Antibiotic susceptibility of fungi, it is resistant to penicillin, tetracycline, and chloramphenicol, whereas in bacteria, it is resistant to griseofulvin and amphotericin B. Dimorphism of fungi is mycelial or yeast form. There is no dimorphism found in bacteria. Chromosomes more than one and con-associated is found in fungi, and in bacteria only one associated. Sedimentation coefficient is 80s in fungi, whereas in bacteria it is 70s. Cultivation is less than 6 pH in fungi, whereas in bacteria it is around 7.2 to 7.6 pH. Morphologic forms. Yeast. This slide focuses on yeast. Yeast are single-celled, usually spherical or oval in shape, measuring around 3 to 15 μm. They grow best at 35 to 37 degrees Celsius, producing colonies that are moist, creamy, and opaque. Resembling bacterial colonies but lacking aerial hyphae, identification is mainly through biochemical testing. Most reproduce by budding, though some divide by binary fission. An example is Cryptococcus neoformans, an important opportunistic pathogen. Yeast are especially relevant in bloodstream and mucosal infection. Molds. Molds are multicellular filamentous fungi. They grow at 25 degrees Celsius, forming colonies that are fluffy, cottony, or powdery. Identification relies on growth rate and microscopic features such as hyphal structure and spore arrangement. Examples shown here include Aspergillus fumigatus, a common airborne mold that can cause severe diseases in immunocompromised patients, Rhizopus species with rhizoids, molds, these showing rhizoids. Molds are the primary source of airborne fungal spores that can be inhaled into the lungs. Dimorphism and polymorphism. Some fungi exhibit dimorphism, the ability to grow as either yeast or mold depending on environmental conditions. Typically, they exist as yeast at body temperature around 37 degrees Celsius with carbon dioxide present, and as mold at room temperature around 22 degrees Celsius to 25 degrees Celsius. Example include Blastomyces dermatitis, Paracoccidioides, Brasiliensis coccidioides, Imitis, Sporothrix, Skansky, Penicillium marnifi, Histoplasma capsulatum, vers capsulatum. So, these are the examples of molds. Polymorphic fungi, both yeast and molds form in the same culture. So, polymorphic fungi display both either yeast and mold form in the same culture as seen with Exophilia species. So, this feature is important in diagnosis because laboratory culture conditions determine fungal appearance. Hyphae. General morphologic features of mold. So, hyphae, the basic structural unit of molds. Hyphae are tubular, branching filaments around 2 to 10 μm wide that make up the fungal body. They may be septate with cross walls or aseptate or coinocytic without cross wall as in zygomycetes. Example here shown: septate hyphae in Phaeoacremonium species and sparsely septate hyphae in mucorales. So, hyphal structures are critical for microscopic identification of fungal species. These are the examples: septate versus aseptate. These species displaying septate hyphae. Mucorales hyphae in tissue appears sparsely septate. First example is septate hypha. Second is coinocytic hypha. Hyphae. So there are three types: antler hyphae, racute hyphae, and spiral hyphae. So these slide describe variation in hyphal morphology that helps in fungal identification. Antler hyphae have swollen branching tips resembling moose antlers seen in Trichophyton. Racute hyphae consist of enlarged club-shaped cells resembling a tennis racket found in Epidermophyton. Spiral hyphae are tightly coiled, corkscrew-like structure seen in Trichophyton mentagrophytes. These unique structural modifications are useful for distinguishing dermatophytes and other molds microscopically. Hyaline versus pheoloid. So hyaline, it highlights the pigmentation differences in hyphae. Hyaline hyphae, that is moniliceous, are non-pigmented or lightly pigmented. Pheloid hyphae, dematicious, are darkly pigmented due to melanin in the cell wall, appearing brown to black in color. Melanin provides resistance to host defenses. The Masson Fonta stain helps differentiate them in tissue. Hyaline hyphae stain pink to red in color, while pheloid hyphae stains brown. Recognizing pigmentation is crucial because dematicious fungi often cause chronic, difficult to treat infections. General morphologic features of mold, that is mycelium. Here the concept of mycelium is introduced. So there are different types: spiral hyphae, pectinate body, favic, chanticleer, antler hyphae, nodular organ, racute hyphae, each given the diagrammatic representation. Here is the microscopic diagram of spiral hyphae. This is antler hyphae and racute hyphae. Mycelium, it is a mass of intervening structure composed of hyphae. It accumulates during the active growth. So vegetative portion or thallus, it grows in or on a substrate and absorbs water and nutrients and anchor the colony. Its reproductive portion or aerial is containing fruiting bodies that produce the conidia and spores. It extends above the agar surface. Fungi reproduction, sexual reproduction. So these slides describe four main types of sexual spores: ascospores, basidiospores, zygospores, and oospores. So ascospores, sexual reproduction, fungi, that is telomorph. It requires formation of special structures so that fertilization or nuclear fission can occur. It gives rise to meiosis, then merging of the cell, and last is nuclear fusion. Asexual reproduction, imperfect fungi or perfect fungi, that is imperfecti anarmorph. It did not exhibit a sexual phase. Spores are produced asexually from mycelium. So ascospores, It is formed inside a sac-like structure called an ascus. For example, these are the examples showing all the phases of meiosis and different types of ascospores. It contained in a sac-like structure called ascus. Following meiosis, 4 to 8 meiosis form with an ascus present. Example is a cleisthothecium of pseudolysaccharid body that has opened and is releasing numerous ascospores. It is a graphical representation of ascospores. Second is basidiospores. It is produced externally on a club-shaped structure called a basidium. For example, mushrooms and cryptococcus. It again, following meiosis, 4 mesospores usually forms on the surface of basidium. Zygospores, thick-walled spores produced by the fusion of two identical hyphal cells, that is, for example, rhizopus. It's rough-walled spores produced by the fusion of two identical cells arising from the same hypha. You can see here in this picture. Last is oospores. It is formed by fusion of non-identical hyphae during heterogeneous fertilization. It is involved fusion of cells from septate to non-identical hyphae. Asexual spores. Here, this slide explains arthoconidia, that is arthrospores. The simplest types of asexual spores, they are formed by fragmentation of hyphae into square or rectangular units. Um, Mature arthoconidia are thick-walled and may align alternately separated by empty or disjunctor cell. Example includes coccidioids, amitis, the cause of coccidiomycosis, and Geotrichum candidum. So arthoconidia are highly infectious because they are easily airborne diseases. These two are the examples. Arthoconidia formation produced by the breaking down of a hyphal strands into individual rectangular units. This is the microscopic image of arthoconidia. Second is blastoconidia. Blastoconidia that forms blastospores are produced by budding from a parent fungal cell. The daughter cell enlarges, buds off and detaches. Sometimes they elongate to form pseudohyphae, a chain-like arrangement seen in Candida species. Many yeast reproduce through these methods. The slide also shows Clastosporium species. These species where blastoconidia are formed in chains, emphasizing how budding is a major reproductive strategy for pathogenic yeast. Chlamydoconidia. So this slide introduced chlamydoconidia, that is chlamydospores, which are thick-walled, resistant resting spores. They form by enlargement and rounding of terminal intercalary or hyphal cells. Their primary role is survival under harsh conditions. Candida albicans is known to produce chlamydoconidia, which aids in identification in the laboratory because of their resilience. These spores allow fungi to persist in the environment for long period of time. Sporangiospores. Sporangiospores are produced inside a closed sac called sporangium, supported by a stalk-like sporangiophore. When mature, the sac ruptures to release spores. This type is characteristic of zygomycetes such as rhizopus and mucor. These fungi often grow rapidly, producing fluffy colonies. This slide image shows sporangia with rhizoids at the base are typical features of rhizopus. Sporangiospores are significant in wound infections and mucormycosis. This is an example, rhizopus species showing sporangium on long sporangiophore arising from pauciseptate hyphae. See here, note the characteristics of rhizoids at the base of sporangiophore. Conidia. So it focuses to conidia, the most important and common type of asexual spores. Conidia are produced either singly or in chain or in clusters from specialized hyphae called conidiospores. They can arise directly from vegetative cell or specialized conidiogenous cells. Two important types of conidiogenous cells are highlighted: Phyalides, which are flask-shaped and produce Phyaloconidia, and annelids, which produce Anneloconidia with ring-like scars. Conidial development can occur through blastic conidiogenesis, that is swelling before septum formation, or thallic conidiogenesis, that is septum forms first, then growth beyond it. So conidia are central to fungal identification because their shape and arrangement are species specific. Examples: blastic conidiogenesis. It appears parent cells enlarges a septum forms and the enlarged portion splits off to form a daughter cell. Thallic conidiogenesis, in septum forms first and then new growth beyond the septum becomes daughter cell. Here is an example: conidia produced on specialized structures. In this preparation of trichophyton species, the numerous small spherical mycoconidia are constructed with large elongated macroconidium. So there are two types of microconidia. First is microconidia and second is macroconidia. Microconidia are small unicellular spores that may be round, oval or pear-shaped. They can be sessile, that means attached directly or on a stalk. That second is macroconidia are larger in shape, multicellular and typically spine or club shaped. They can be smooth walled or spiny. An example shown here is Trichophyton species where numerous conidia, that is microconidia, contrasts with larger macroconidia. These structures are key in differentiating dermatophytes in clinical mycology. Example of Trichophyton mentagrophytes, microscopic images you can see here. Phyaloconidia. So this slide focuses specifically on Phyaloconidia produced from a flask-shaped cells called phyalides. In Aspergillus, long chains of rough wall conidia are produced with youngest conidia at the base and mature ones at the tip. In Acremonium, conidia are held together in slimy mass. In Penicillium, Filiates arranged in a brush-like structure give rise to chain of conidia. The morphology of filiates and conidial arrangement is a hallmark of fungal taxonomy. So, conidiophores form a penicillus that determines in secondary branches, that is metulae, and filiates from which chain of conidia are born. Here is an example of penicillium. Flask-shaped filiates giving rise to long chains of conidia, characteristics which is a characteristic of Aspergilli. The basal conidia are the youngest one, whereas mature conidia have rough walls. You can see in 400x. Chain of conidia was produced by annelide. Classification of fungi. First is through botanical class taxonomy and second is type of mycosis. So, botanical taxonomy: Zygomycota reproduce via zygospores and sporangiospores. Ascomycota produce ascospores sexually and conidia asexually. Basidiomycota reproduce sexually via basidiospores. Deuteromycota, it is fungi imperfect. It lack a known sexual state, reproduce only by conidia. Additional fungi are classified by the site of infection, that is superficial and cutaneous mycosis, subcutaneous, systemic, and last is opportunistic mycosis. First is Zygomycota. So they produce hyaline sparsely septate hyphae. Asexual reproduction occurs through sporangiospores, while sexual reproduction produces zygospores. Clinically important genera are Rhizopus, Mucor, Rhizomucor, Lithemia, formerly Apsidia, and last is Cunning Gamelia. Here is the sexual reproduction and asexual reproduction of Zygomycota. Second is Ascomycota. They reproduce septate hyphae and reproduce asexually via conidia and sexually via ascospores. Many clinically important fungi belong here, that is dermatophytes, pseudolos body, histoplasma capsulatum, aspergillus. These fungi are widespread in the environment and include both pathogenic and opportunistic species. It is ubiquitous in nature and produce true septate hyphae. All exhibit sexual form but also exhibit asexual form. Microsporum species include in Ascomycota. All the species included in Ascomycota. This is a sexual reproduction and asexual reproduction cycle of Ascomycota. Basidiomycota. They reproduce sexually by forming basidiospores on club-shaped basidia. They often form clamp connections at septa in vegetative hyphae, which helps in identification. Most are plant pathogens or environmental fungi, but some can infect humans. The most important human pathogen is Cryptococcus neoformans complex, which causes cryptococcisis. Its telemorph is Phylobasidilia neoformans. Other examples include mushrooms, rust, and smuts. So, this is the sexual reproduction and asexual reproduction of Basidiomycota. Deuteromycota. They are not a natural taxonomic group but an artificial one. Deuteromycota, also called as fungi imperfect, it is created for fungi without a known sexual stage. They reproduce only asexually by producing conidia. Most resemble is compared to Ascomycota. Example include epidermophyton, dermatophyte, and sporothrix. This category has become less important with advances in molecular biology, which help identify the true phylogenetic position of this fungi. Comparison of the fungal phyla important to human diseases. Phylum Zygomycota. It is common name is Zygomycetes. Crosswall septa is absent in Zygomycota. Sexual structure is zygospores. Sexual spore is again zygospores. Asexual spore is sporangiospores. Representative genera of Zygomycota is Rhizopus. Second is Ascomycota. Its common name is Ascomycetes or sac fungi. Septate is absent in Ascomycota. Sexual structure is ascus, whereas sexual spore is ascospores. Asexual spore is conidia. Representative genera is Saccharomyces, Aspergillus, and Penicillium. Last is Basidiomycota. Common name is Basidomycetes. It is a club fungi. Crosswall, that is septa is present in Basidiomycota. Sexual structure is basidium, whereas sexual spore is basidiospores. Asexual spore is conidia, that is hyphal fragments, and its representative genera is Agaricus and Amanita. Zygomycota, its growth characteristic is non-septate hyphae. Asexual spore type is sporangiospores. Human pathogens include Rhizopus and Mucor. Its habitat is ubiquitous. Type of mycosis is systemic. Clinical notes, that is opportunistic pathogen. Ascomycota, its characteristic is dimorphic in nature. Asexual spores is conidia. Human pathogens include Aspergillus, Blastomyces, Dermatitis, Histoplasma, Capsulatum. Its habitat is ubiquitous for Aspergillus. For Basidomycetes, it is still unknown. And for Histoplasma, soil. Types of mycosis for Ascomycota, Aspergillus is systemic. Blastomyces is again systemic, as well systemic for histoplasma. Clinical notes: opportunistic pathogen Aspergillus, inhalation through blastomyces. For histoplasma also inhalation. Growth characteristic for Ascomycota: it is septate hyphae and exhibits strong affinity for keratin. Its asexual spore type is Conidia. Microsporum human pathogen. Habitat includes soil, animals. Type of mycosis is cutaneous. Clinical notes is Tinea capitatis, that is ringworm. For Arthoconidia, asexual spore, its habitat is again soil and animals. Type of mycosis is cutaneous, and clinical notes is Tinea pedis, that is athlete's foot. Enormous septate hyphae. You can refer this table for enormous and basidiomycota. Mycosis. So mycosis. This slide introduces the clinical classification of fungal diseases, that is mycosis. Superficial or cutaneous mycosis. It involves keratinized tissues like hair, skin, and nails. Example include dermatophytes, that is ringworm, Tinea nigra, Tinea versicolor, and pedria. These are the examples. Subcutaneous mycosis, it involves deeper layer of skin, muscle, and connective tissue, often following trauma. Example includes pyrotrichosis, mycetoma, chromboblastomycosis, and phaeohemicomycosis. Systemic mycosis primarily affect the lungs but may disseminate to other organs. Example include blastomycosis, coccidomycosis, histoplasmosis, and paracoccidomycosis and penicillosis. Opportunistic mycosis. It occurs in immunocompromised individuals or person having underlying health issues. Examples are aspergillosis, candidiasis, cryptococcosis, and zygomycosis. Superficial or cutaneous mycosis. So this slide focuses on this. These mycoses are restricted to the outermost keratinized layer of skin, hair, and nail. They do not invade deeper tissues. The main causative organism are dermatophytes. Example, Trichophyton, Microsporum, Epidermophyton, and yeast-causing conditions like Tinea versicolor, Tinea nigra, and pedria, black pedria, and white pedra. Clinically, these infections are characterized by scaling, discoloration, or nodules, but are usually not life-threatening. Subcutaneous mycosis involve infections that penetrate deeper into the skin, subcutaneous tissues, and sometimes bone, usually through trauma such as thorn, pricks, or cuts. Unlike superficial infection, these diseases are more chronic and localized. Example includes sporothrix, rose gardener's disease, mycetoma, chronic granules infection causing discharging sinuses with grains, chromboblastomycosis, caused by dematicious fungi producing sclerotic bodies in tissue. Phaeohyphomycosis, another dermatitious fungal infection with pigmented hyphae in tissues. These diseases typically require prolonged antifungal treatment or surgical removal. Endemic primary systemic mycosis. So, systemic fungal infection which primarily begin in the lungs after inhalation of fungal spores. These infections may remain localized or disseminate to multiple organs. Systemic mycosis are caused by dimorphic fungi such as blastomycete dermatitis caused by blastomycosis. Histoplasma capsulatum causes histoplasmosis. Coccidio Immitis causes coccidioidomycosis, that is valley fever. Penicillium causes penicilliosis, especially in HIV patients. These diseases can affect both immunocompetent and immunocompromised individuals. Opportunistic mycosis occurs in individuals with impaired immunity, such as those which HIV and AIDS, cancer chemotherapy, organ transplant, or prolonged antibiotic therapy. Common opportunistic fungi include Candida species, that caused by Candida albicans and other Candida species, Aspergillus species, Aspergillosis, Aspergillus fumigatus, and other Aspergillus species, Halohyphomycosis caused by species of Fusarium, Pseudomyces, Trichosporon, and other hyaline molds, Pheohyphomycosis caused by Cladophilero, for Bantatina species of Alternaria, Cladosporium, Bipolaricis, and numerous other dematicious molds. Mucormycosis, that is zygomycosis, caused by species of Rhizopus, Apsidia, and other zygomycetes. Penicilliosis, caused by causative fungal agent, that is Penicillium burnetii. So, summary. The final, it It provides an overview and conclusion how it emphasizes the medical mycology, which is the study of fungi causing human diseases ranging from superficial infection to life-threatening systemic mycosis. Fungi may be true pathogens, that is infecting healthy hosts, or opportunistic pathogens affecting immunocompromised patients. Their identifications relies on morphology, reproduction, pigmentation, and laboratory diagnosis. So its definition, what is uh, mycology? Meaning study of fungi causing human diseases, type of pathogens. True pathogens infect only healthy individuals, whereas opportunistic pathogen affect immunocompromised or immunocompetent patients. Fungal forms, yeast include unicellular, that is example of Candida. Molds, multicellular hyphae caused by Aspergillus. Dimorphic fungi and mold, blastomyces, histoplasma are the examples of molds. Classification, zygomycota, which include rhizopus and mucor. Ascomycota, it includes dermatophytes, histoplasma, and Aspergillus species. Basidiomycota, it includes Cryptococcus species. And Deuteromycota, sporothrix is an example, which including Deuteromycota. Mycosis and diagnosis. Clinical types of mycosis: superficial cutaneous, which includes skin, hairs, and nails; subcutaneous, deeper tissues; systemic, that is dimorphic, which include lungs and dissemination of histoplasma, blastomycosis, and coccidioides. Opportunistic includes immunocompromised, Candida and Aspergillus species, Cryptococcus and Zygomycetes. Its lab diagnosis include direct microscopy, culture, histopathology, serology, and molecular test. Questioning part. So for 3-mark questions, there is 5 questions. First is define medical mycology and give 2 examples of each for pathogenic fungi. Second is what are dimorphic fungi? Write 2 examples. Third, name the 3 genera of dermatophytes. Fourth, what is the diagnostic features of Cryptococcus neoformans? Fifth, mention 2 opportunistic fungal infection, that is Candida and Aspergillus. For 5-mark questions, write a note on the general characteristics of fungi. With example, you can include. Second is describe the classification of mycosis with example. Third, write short notes on dermatophytes, that is ringworm infections. Fourth, discuss the clinical features and laboratory diagnosis of histoplasmosis. And last is write a note on mucormycosis, that is zygomycosis. So that's all for part 1 of mycology. In second part, we'll study in detail all the diseases, for example, candida, all these diseases in detail, which is caused through fungal infection.
Yeah, very nicely covered, madam, regarding fungi. Okay.
Mycology, fungi, yes, sir.
So the terminologies which are
The,
used in fungus.
yes, sir. Fungus.
So our students has to
Hmm.
repeatedly they have to undergo these words
This,
actually.
correct,
So then
sir.
only the terminologies they can remind.
They
Otherwise,
with
they can forget easily.
forget this with this terminology, they can differentiate what and all include in which category.
Yeah,
For
yeah, yeah.
mold and yeast, it will include in dimorphic fungi. Like this, they'll get to know.
Yeah. Very nicely explained, madam. Thank you. Thank you
Thank
so
you.
much.
Thank you, sir.
Yeah. So next we will start at 6
Mycology
o'clock.
part 2. In
Yeah.
detail, we'll study about all the diseases, sir.
Yeah. 20 minutes we can have some break.
Yes,
So
sir.
we can rejoin.
Thank you,
Yeah.
sir.
Thank you.
Thank you, sir.